All right, folks, let's get started. Thank you all for coming. It is my pleasure to introduce our seminar speaker today, Dr. Douglas Walker. We tried to ask him to come last semester, and we were not able to. He was not able to make it here, but this year, this semester, we were able to bring him here. Dr. Walker's originally trained in London for his undergraduate in BTech in applied biology, so you can say that technology BTech is pretty smart degree. So he did the applied biology, then went to graduate work in University of British Columbia, one of the beautiful campuses in the West Coast. He did his graduate work, and then went to Georgetown for a postdoc for a couple of years, and then went back to postdoctoral work in some top neuroinflammation people in the UBC campus again, and then got recruited to Sun Health Banner Institute over 15 years ago?
18
years ago. Then recently he's also affiliated with, and he moved to his research lab a little over a year ago to Arizona State. Dr. Walker's work is pretty impressive. The good things about his work is he uses a real model. That's the human samples. Well, some of the basic scientists in this campus in many places, we always fiddle with cells and animals, but he works in real samples. And the other thing always I like about his work is the detail. Some of the things to do with inflammation, some of the new concepts. So it's a very impressive type of work, and he's going to share with today about some of his findings that he's done over the years, some of the new work on the Alzheimer's and aging, and the microglial activation and inflammatory signaling. So we're lucky to have Dr. Walker. All yours.
Thank you. Okay, good afternoon. It's a great pleasure to be here. Yeah, I was 18 years at the Banner Sun Health Research Institute, but a year and a half ago they came up with the idea of sort of joining us with the Arizona State University. So I've only been a professor for a year, so it's a new experience. 30 years ago, I was a postdoc in 1986 in Vancouver. This was kind of like the 30-year anniversary of what we call the modern generation of neuroinflammation. In October 1986, they started looking at microglia. Pat McGeer and their group had many different Japanese colleagues, but my background was actually in virology, so I was sort of hired to look for herpes viruses in Alzheimer's, but it didn't really pan out. So with the 30-year mark, I thought instead of having just a pure biochemical, I'm going to give you a few opinions about the state of neuroinflammation in aging, how maybe some of the concepts that have been developed need to be re-examined, and I'll use some of my own work, which is a little arrogant, but to sort of justify my ideas and sort of maybe sort of suggest where the field could be going. So as we all know, Alzheimer's disease, most of my work's on Alzheimer's, but some of this will be dealing with Parkinson's as well. Alzheimer's disease is a serious degenerative disease of the elderly. These are the stats for estimated for 2016. Obviously, the years, so these are estimated. But the feature to notice is how it is primarily a disease of aging. If you get it before 65, it's usually a genetic early onset Alzheimer's disease. But the numbers grow as you get older. 75 to 84 is probably the peak time. And once you make it past 85, and they even say if you make it into your 90s, you're probably resistant to Alzheimer's. It's now being appreciated as a serious cause of death. Now this could, you see these changes in death certificate figures for the common diseases, breast cancer, prostate cancer, heart disease, HIV. But there's been a huge increase in the physicians' understanding of how serious cause of morbidity or mortality Alzheimer's disease is, a 71% increase. This is probably due to the physicians previously diagnosing the patient for the reason that they died, which could be sort of a general infection. So when you consider Alzheimer's disease, there's a lot of popular journalism written about it. And so if you look at, I thought this was interesting, and it sort of also highlights that mainly a lot of scientists have failed. So in 2000, you have this cover, New Science of Alzheimer's Disease. And then you have 6 years ago, at least some progress against this disease is being made. And now, 2016, the Alzheimer's pill, which is probably still wishful thinking. But I was taken by a comment I just read the other day. It was by Rudy Tanzi, who's the professor of neurology at Harvard. He's famous for his work in discovering the amyloid precursor protein. This was actually written in the AARP bulletin, so it's designed. And so the take-home message is it's really inflammation that takes you out. The plaques and tangles push you up the mountain, but you could live on top of that mountain forever if there's no inflammation. I'm not quite sure what that analogy means, but I agree with the message. It is the inflammation that results in the plaques and tangles that sets the brain on fire. So when you consider Alzheimer's disease, I've been sort of working with an autopsy cases for many years. This is sort of like a stock picture, but the feature is you get severe gross atrophy, particularly of the frontal regions, the temporal regions, and the ventricles. Now, this picture shows actually a concept that I'll be getting back to is the spread of the disease. So in early cases, people who, most elderly cases, this is a Bilchowski stain, silver stain, to show the neurofibrillary tangles in the hippocampus and the entorhinal cortex. But as the disease progresses, it spreads further. And so it's a spreading disease. And as you imagine, a lot of the parts of the brain that earlier were not affected, so the hippocampus can be considered like a junction box. Further, as it moves into the cortex, you get the loss of cognition and memory functions, and this gets deeper and deeper. So this was it had a severe human effect. This is Alzheimer's 1905 in his index patient. The picture on the right was actually my mother, who died of Alzheimer's last year, and you can see it takes a severe toll on the families. So as an Alzheimer's scientist, you know, having had this affected in the family, we realize that we still have a lot of work to do to find a proper cure. So this is the background stock slide. Alzheimer's disease research for the last few years has focused on amyloid precursor protein and how it can make plaques. The amyloid precursor protein is about 130 kilodaltons, a transmembrane protein, but when it gets cut in a particular manner or processed, you get the formation of either the alpha secretase cutting, so you get a soluble form which is released, but if in pathological processing, you get the cuts by beta secretase and gamma secretase forming the widely known amyloid beta peptide. This peptide, because it's crossing the transmembrane domain, has lots of hydrophobic regions, so it has a tendency to aggregate. And so for many years, and even now, this has been the focus of how to cure Alzheimer's disease. Whether this has been correct or not, it still remains to be seen whether this should be the primary focus or just a focus. As I said, I've had my opinions. Another feature is also the reason you get a buildup of these senile plaques is there appears to be a failure in the A-beta degradation processes. So amyloid is made from birth, but then you only get the buildup with age. So it's also hypothesized that enzymes like neprilysin, matrix metalloproteinase 9, and IDE, insulin-degrading enzyme, might be defective. There's good evidence for this. So the clinician's real goal is to try and identify Alzheimer's early so that you can maybe treat it early, even though there's not really effective ways of treating it yet. This is kind of the panel of what we call the biomarkers that are being carried out preclinical, MCI, and in dementia. So usually the preclinical data would come from volunteers from studies. MCI basically is considered by some as pre-Alzheimer's. You start to show some cognitive decline, but not enough to meet the criteria of dementia. So in terms of biomarkers, right now, A-beta in the CSF or with a positron emission tomography scan is the #1 feature, and it seems to have the sensitivity. You start to see early changes. The trouble with this test, if you're doing in CSF, is you actually are looking for a decline, which could cause scientific problems. PET scanning basically is looking for accumulations of plaques in different regions of the brain. An earlier study, which also points to different types of mechanisms, is the glucose metabolism. You can do PET scans for fluorodeoxyglucose, and so you actually see a decrease in glucose metabolism in Alzheimer's brain, which can actually be picked up early. Now, tau in the CSF, tau is a microtubule protein that is also become phosphorylated in Alzheimer's disease, and so it becomes abnormal. It also aggregates. There's been a large controversy about which comes first, the A-beta or the tau, in terms of the pathology progression mechanisms. If you measure tau in the CSF, it actually starts to go up progressively as the disease becomes more severe. And you can imagine as neurons die or they become abnormal, the tau is being released into the cerebrospinal fluid. So these are the two diagnostic biomarkers now. If you do them with CSF, they are reasonably cheap, but they have, the literature is full of discordant results about what do they actually mean. And, and also, can you say if you have a tau level of, say, 600 picograms per mil, that means you've got Alzheimer's or not. So there's a lot of controversy. But besides the basic scientists, you've also got people looking at magnetic resonance imaging, so you can actually start to see in living patients the brain atrophy. This comes later than some of the biochemical changes. And then usually then you move into the realm of the neuropsychologists, where you actually start to see cognition. There's been a lot of controversy. If an elderly person goes to their regular physician, they will have the physician can do what's called a mini mental status exam. It has a kind of inaccuracy, it's sort of kind of loose, but when they start to become research subjects, and the proper trained neuropsychologists, you can have a whole panel of cognition tests to actually, it turns out to be a very good predictor of dementia. But what can happen is in Alzheimer's disease, dementia does not predict Alzheimer's disease. Dementia predicts basically loss of function. So you can have other features like Lewy body dementia, possibly strokes, medication. So there's a need for all these biomarkers to work together. And then, finally, once you move into dementia, this is also amazingly hard still for a lot of physicians to diagnose, but they only see the patient for 10, 15 minutes. What in our field has been a particular interest is to see where does inflammation occur in this story. So inflammation has been considered a consequence of the plaques or maybe a driver of the plaques. So this basically is to highlight the need for new therapies. There's quite a large deficit in possible targets. So Alzheimer's therapy is mainly focused on amyloid. But this is a list of current and past FDA-approved trials based on amyloid-related therapies. And so right now, you have a very few active. These are all going to be focused on Alzheimer's amyloid immunotherapy. A lot of them have failed. You've got 13 out of the 50 have failed, and some have basically become inactive. But I'm kind of disappointed, and this is where, again, as a basic scientist, we've kind of failed to come up with better targets for inflammation. Back originally, the idea of treating Alzheimer's disease with an anti-inflammatory was taken off, and they did many trials. But right now, if you look through the list, if you go deeper into this data, you'll find actually the targets for inflammation that people are trying to study are very few. We're still looking at the steroids or the non-steroidal anti-inflammatories. So we, as basic scientists, still need to sort of further understand some of the concepts of, what are the mechanisms of Alzheimer's inflammation. So, briefly now, the major feature are a number of antibodies, what's called passive immunization with humanized monoclonals. A number of these large drug companies are spending billions on these antibodies. They offer great hope of maybe you inject them into the patient, they bind to the amyloid, and some of them make it across the blood-brain barrier, and they actually help remove the amyloid. And so some of these trials have failed, some are still ongoing. We'll have to probably wait another year or two to find out if this is going to be the treatment. There's been a lot of damage as things go on, as many trials have failed. A lot of the other trials also, related approaches are intravenous immunoglobulin, which basically, you take human serum and you see if you've got anti-amyloid antibodies, and then beta and gamma secretase inhibitors. These are to stop the A-beta being formed in the first place. These are still ongoing. The gamma secretase inhibitors generally have not worked because they have serious side effects. One of the troubles is when you start to inhibit many of these enzymes, you also are inhibiting normal cellular processes. There's great hope for beta secretase inhibitors, but again, this is unknown. Again, whether they will actually be effective in reversing cognition deficits. So, as a basic scientist, you can sort of approach Alzheimer's and inflammation, neurodegeneration, and sort of from several angles. You can, first of all, look at the human brain. I've been fortunate enough to be able to study the human brain, have samples, so you actually know what's going on in the human brain. And then also, as I will describe, you'll have cells that you can grow from the human brain, the microglia. This is a picture of microglia stained with the antibody IBA1. But ultimately, any targets, any mechanisms have to be tested in animal models. I don't do that much type of that work. It's sort of, we focus primarily on the human neuropathology, the human cells. But ultimately, the goal is to find a therapy that can actually affect the brain of the elderly person. So, so I'm going to discuss sort of the neuropathology and the cell culture approach. This slide should probably have been earlier. It's sort of kind of like the classical observation. This is the microglia, the brain-resident macrophages. That's a simplification. They are not just macrophages. They are highly specialized cells with a macrophage lineage. So in an elderly non-demented brain, you see these cells. Their job is basically housekeeping. And so they have these branches. These are stained with HLA-DR, the MHC2 protein, that is very, very widely used. But as I'm going to explain, I think has limitations. When you look at an Alzheimer's case, the thing you notice is you've got more stained cells, and then you've also got very dark stained clumps of cells. And so this has been taken to indicate that the microglia are activated. And that is a term that really has to be defined very carefully to be meaningful. But this is a kind of like a hallmark feature of neuroinflammation and Alzheimer's disease. The microglia respond to the amyloid plaques. They are activated by the plaques. They're trying to eat the plaques. But what has been believed is that when these cells become activated, they can produce lots of inflammatory toxins, tumor necrosis factor, reactive oxygen species, superoxides, free radicals, many toxic enzymes. And so this has been the idea that why inflammation could be causing downstream effects or accelerating Alzheimer's disease. It is a theory that is probably true, but it has to be sort of refined in order now to be able to be effective in treatment, because drug trials for steroids, the most widely known anti-inflammatories, are ineffective and have serious side effects. I quote this paper just for the historical reasons I was mentioning. This was the first paper. It came out of work just after I joined the lab. McGee's. I'm not a co-author. I was just there at the time witnessing history. But because of that work, and I was sort of able to carry on in the lab for a number of years. I just wanted, because of the interest of the host, just to mention our work on Parkinson's disease. You do see similar features of activated microglia. These are the purple ones. These are in the substantia nigra. The thing I had this in a recent paper, is the fact that you will find that even in control cases, these are non-Parkinson's cases, this is a substantia nigra, so you've got the neuromelanin cells. You've got, you've got a certain number of activated microglia, but then when you start to have Parkinson's disease, you've got a much, much more battlefield. The interesting point is that the substantia nigra itself in humans is a highly inflammatory environment. And so with aging, all these samples will be from aging people, 80, 85-year-olds. So you can see aging has an effect on inflammation too. We'll discuss that. So getting back to the focus of the talk, I'm going to be mentioning sort of the feature of the research is endogenous anti-inflammatory system. How does the brain protect itself, and are these possible targets? We're going to mention CD200 and CD200R. These are sort of molecules that affect the interaction of microglia and neurons. CD33, which has had a genetic association, and also a new study that we are looking at, the possible interaction of sugar and Alzheimer's disease. Our experimental system, again, this is sort of the heart of this, is that we, at the Sun Health Research Institute, they collect the brain from donors 2 to 3 hours after death. They are sliced, and we are given some tissue, and from these we can actually grow different types of cells, everything except neurons. We've got microglia, we've got astrocytes, we've got smooth muscle cells, pericytes, and endothelial cells. The focus will going to be on the microglia. I just put this in just for vanity, but a lot of my work has been criticized, or people who do work with microglia gets criticized because they say once you take them out of the brain, they basically become macrophages. And so we've done careful studies over the years, so you can see microglia have these processes. You look back on the picture that I showed a couple of slides ago, they've got processes, and you can see this in culture. And so even after 5 weeks in culture, that's a bit dark, but it shows up. It's a phase contrast picture. You still have the microglial processor. And so in recent years, people are saying, what is a microglia? And so they're trying to find markers. So people can take the microglia directly out of the brain and do this sort of analysis. We were doing this with cultured cells. So we had the cultured microglia and the cultured macrophages. And we were very happy to see that what has been established by people, P2RY12, a purinergic receptor, and a membrane receptor, are highly overexpressed or in our microglia even after 2 weeks compared with the macrophages. So we are satisfied that they are still microglia even though they've been cultured. A lot of people will disagree with me. So obviously our goal is to identify therapeutic targets, and we're using descriptive human brain studies. So we're looking at CD200, CD33, a process called oblique inoculation. And if I don't run out of time, I also want to discuss some of the work of how you subtype microglia. So the question is, what are the concepts of inflammation in neuropathology? The idea of M1 and M2 microglia is being established by immunologist Simon Gordon at Oxford, a famous microglia person, and sort of kind of says they don't exist. It's an interesting article, very provocative, as is. He tends to be provocative, but I think I would disagree with that, but we have to sort of show that he's wrong. So with CD200, essentially what you have is you've got CD200 is a molecule on neurons, and it binds to a molecule called CD200 receptor on the microglia, and it activates anti-inflammatory signaling. That's kind of like the Cliff Notes. It's expressed by neurons. So this has become one of the major systems for the body in many different organs to turn off inflammation, an endogenous anti-inflammatory. And in cancer, it's been shown to actually be related to, if a cancer cell is overexpressing CD200, it actually has an increased chance of metastasis. So this is actually can be good in some systems and bad in other systems. So the work we started a couple of years ago was to actually look at CD200 in human brain. This was the first human brain study. We actually produced our own antibody. The commercial ones didn't work. So what we were seeing, again, with CD200 is it's primarily in neurons, but we also saw it in a subset of astrocytes, but not in microglia. So there's you see there's no co-localization of the CD200 with the microglia, just with the neurons. So we were using traditional methods of analyzing. So we took hippocampus, inferior temporal cortex, both regions severely affected by Alzheimer's disease, and cerebellum, which is like our control. And so we were able to show significant decreases in CD200 mRNA in the hippocampus and the inferior temporal cortex, but not the cerebellum, both at the mRNA level and the protein level. Surprisingly, we also saw similar decreases in CD200 receptors. So this system has got severe deficits as the Alzheimer's progresses. What we were studying, though, was elderly people. And so one of the problems with a brain bank with elderly is that you can't have young people. Getting hold of younger brains is a problem. But luckily, I was working with some good people who have friends in the wrong places so they can get other brain samples. So with my colleague Alex Roa, we did two studies. He was comparing young adults, and then as they get progressively older and with disease, and a separate cohort of young, middle-aged, and old. And this nicely confirmed, these studies were done at the protein level for looking at CD200 decrease with aging. This is kind of typical of human studies, is you get very high variability. And so it is sort of a feature that you have to sort of live with, and sometimes your statistics appear a little bit weak. So our work confirmed earlier studies by Lyon and Frank, basically showing CD200 goes down with age, and we were able to confirm, again, that you have a further deficit with disease. And recently, we got hold of some gene expression data from a UC Irvine Banner Sun Health Research Institute data set. So we were able to show in both hippocampus and SFG, you got young, middle-aged, old, MCI, and you see this steady decrease of CD200 with both age and then further with Alzheimer's disease. So you've got two effects. So if you've lost a protective system as you age, this kind of opens you up to potential enhanced inflammation. So we've always asked the question, does age-associated loss lead to inflammaging? It's a kind of cliched term to sort of explain enhanced as aging inflammation. What we had also done is actually, this is kind of, I just briefly discussed this, is to define the mechanism of CD200 receptor. People have done work with rodents, and this sort of highlights the essential part of validating your work with some human materials if you can. As I said, we were fortunate to be able to use pure human microglia. And we were able to show that CD200 receptor was an M2 alternative activation, non-pathogenic marker for microglia. It's turned on by the enzymes IL-4 and IL-13. And you see many of the other common cytokines or inflammation-inducing agents had no effect or no significant effect. Interestingly, lipopolysaccharide and IL-1 beta had an inhibitory effect. So when you have an inflammatory environment, you actually also, if you're turning off CD200 receptor, you're actually pivoting the system against itself by sort of deactivating it. What I wanted to also explore was how does inflammation affect Lewy body pathology? Now, we can talk about Parkinson's disease, but Parkinson's disease is really just a spectrum of alpha-synucleinopathies. A colleague of mine, Dr. Beach, developed a scheme. He published it in 2009 to actually stage the progression of the alpha-synuclein. And this is a complicated scheme, but basically it highlights the features that in many cases you can get alpha-synucleinopathy starting in the olfactory bulb, which is an interesting thing in itself, and then it spreads through the brain into the limbic region, brain stem, which includes the substantia nigra, and then passes on into the limbic system and the neocortical system. So this is a measure of in two features, the stage 4 and the stage 3. And so he developed this scheme, and you can see a progressive increase in the amount of, this is phosphorylated alpha-synuclein, the abnormal form of synuclein, the aggregated form of synuclein. And we showed it in biochemically in a sort of an accompanying paper. You get increased amounts of abnormal synuclein as the disease progresses, and also increased amounts of aggregated synuclein. So the question is, can we use these samples that we studied here? We had about 140 samples to actually look at inflammation in Lewy body diseases. I'm going to skip over this. It's, what we had was groups that were staged with different progressions of disease. We had 143 samples. They were classified according to Lewy body score. The pathology department has its own scheme of ranking. So you look at 10 brain regions and you rank 0 to 4. So you can get a score of 40 if you to reflect the brain load of alpha-synuclein. In the individual areas, it's sort of 0 to 4. So this represents the load in the synuclein, in the cingulate cortex, and the load in the temporal cortex. But unfortunately, with human disease work, you also have other pathologies. So plaques and tangles, these were actually the same scores coming from the pathology department, part of their routine study. And they showed that many of these cases, these are elderly cases, average age 84 to about 85, have significant amounts of Alzheimer's pathology, even though they are not Alzheimer's disease cases. And so we wanted to sort of see if we could use these cases to define inflammatory changes. So we were using CD200 as one marker, and this was to see the protective effect. And we sort of compared it to ICAM, intercellular adhesion molecule, as the other marker, just to see how the changes in these with Lewy body staging. So what happened is in stage 1 and stage 2B, these are cases with mild to moderate, very mild actually, alpha-synucleinopathies. You do see a significant effect. But in the cases with the severe alpha-synucleinopathies, you do not see an effect. So what we did was actually, it was interesting to find out that if you compare the load of Alzheimer's-like pathology to synuclein pathology, what we came up with was the fact was that the changes in these inflammatory markers in both temporal cortex and synucleinopathy did not correlate in any way with the degree of Lewy body pathology. But they did correlate quite strongly with the amount of Alzheimer's pathology. We just submitted this paper for work. It's probably going to be a little bit controversial, and it does have some limitations, but we sort of think it's a concept that was worth putting out there. These are the correlation graphs to show the degree of correlation between the two measures. And this is the correlation to show lack of correlation in temporal cortex for CD200 and ICAM. And the same thing was in a cingulate cortex. I did this recently for the paper, but it was quite surprising was actually when you start to look at the microglia, these are in brown with the purple, which is the abnormal synuclein. What we were actually finding was there was very little co-localization or apparently microglial response to this phosphorylated alpha-synuclein. So we got the Lewy bodies, we got the Lewy neurites. I'm showing these top two panels. These are actually cases that were control cases without Lewy body pathology, but this kind of shows in the elderly that there is a background level of activated microglia in all cases. Now, if you compare that to this is a section, the brown is the microglia, and the purple is phosphorylated tau, the tangle marker. And here you've got the brown microglia, and you've got the plaques. So you see this very strong response to the amyloid plaques and also to the tau structure. These are probably neuritic plaques, so there might be a mixture. So we thought that was kind of interesting. But so again, it sort of highlights a possible different concept of what's going on in the brain in between the two diseases. We also sort of started to show that there was an interaction between different inflammatory molecules. TREM2 is one of the genetic markers that have been studied in relation to inflammation and Alzheimer's disease. And so the correlation, as inflammation goes up and aging, the CD200 and the CD200R goes down. This again was the data set I measured. What was also interesting was if you look at the same results for synaptophysin, the marker for synapses, you can see you have a positive correlation. The more CD200, the more synaptophysin. Again, this is in the same group of cases, the correlation analysis. You got high significance. So the other topic is the role of genes in inflammation. TREM2 has been widely studied. CD33. These are polymorphisms that have been associated with altered genetic risks of Alzheimer's disease. They change the risk factor if you've got a particular polymorphism. So for CD33, you've got a polymorphism in the non-coding region. CD33 is a sialic acid binding protein. It's actually an anti-inflammatory protein in itself, but it's also involved in phagocytosis. TREM2 is a proper mutation within the coding region. We screened all the cases in our brain bank, and we also saw we were able to type the different genotypes for CD33, but and we had quite a few of the different genotypes. The AA is a rare one, but TREM2, there was only one case out of like 1,000. So this work, when it was underway, was also, I was competing it and with Rudy Tanzi because he had a neuron paper before we could get ours published. Our results are similar but not identical, but what was interesting is that we are studying the effect of the polymorphism on the mRNA expression, and we showed actually there was no effect or no significant effect of the polymorphism on the expression of the mRNA. There is a disease effect. But then if you look at the protein, there is a significant effect. The polymorphism quite significantly affects the expression of the protein. And it doesn't matter whether it's an Alzheimer's case or not. These are combined data. So this was just a representative Western blot. You've got a CA case, so that was sort of, and the AA cases are the ones that are considered protective for Alzheimer's disease. And so you can see actually, even in this representative image, there was less CD33 protein in these brain samples. These are brain samples from the temporal cortex that are affected by Alzheimer's disease. And there's quite a large difference. So, and you can do the stats on that. These are just sort of semi-quantitative Western blots. And so you see a significant difference. What was interesting also as a new feature was the fact is that if you measure the level of IBA in these same samples, you got a reverse effect, a significant increase. This could be indication of actually the AA cells are more activated or they're more microglia. But what was different from our results compared to some other publishers, that we could not see a protein difference in these cases between AD and ND. Again, CD33 was studied in our human microglia. This was an immunohistochemistry panel. So you can see it basically will recognize most microglia in a tissue section. You know, the activation state's not known. But what we were surprised to find is that if you activate the microglia in culture with a whole range of activating agents, except interferon gamma, you actually see a decrease. So what you see in culture wasn't quite reflecting what you were seeing in the brain. We're still trying to work out the temporal course of these things. And again, we have data to show that, and this is a concept that needs to be investigated, how they two things interact with each other. Now, another feature, and again, I'm just going to skip over this, is the idea that sugar and diabetes might actually interfere with, have an effect on Alzheimer's disease. There's a process called oglycnacylation, which is the addition of a oglycnac, a galactosamine molecule on proteins. This is like the reverse of phosphorylation. So if you have a protein that's highly glycnaculated, is to use the verb, it's how you have reduced activation. And if it becomes, the same protein becomes phosphorylated, you could have increased activation or aggregation. So this has become an important topic because people are showing that tau in Alzheimer's brain has a loss of oglycnac. And so the idea now is, can you use oglycnac inhibitors, or oglycnac removal inhibitors, to enhance the amount of tau as a therapeutic strategy? So just to recap, what you're happening is that if you, in Alzheimer's disease, tau becomes extensively modified by phosphate groups. But also, some of these phosphate moieties can also be oglycnaculated. So if you have one or the other, the more oglycnac or the more galactosamine on the molecule, the less likely it is to phosphorylate. And so we were able to show that actually, if you could, this also has an effect on inflammation. This was a feature of a grant I unfortunately was not well received, is to show that we could show that inflammation affects the level of oglycnacylation and the markers of the two key enzymes, OGA and OGT. But in our culture system, you also could see that if you treat them with the thiamet G, which is an oglycnac A's inhibitor, so you'd have more oglycnac molecules in the cell. You actually get reduced inflammation. It wasn't tremendous, but it sort of shows that it has a potential. The only drawback is that we were starting to see, if you look in the brain again as opposed to the culture, we were starting to see a profile, NF-kappa B as a transcription factor. And it's widely studied because it's a key role in inflammation. And a lot of it's been studied, oglycnac, in diabetes. So in diabetes, when you've got too much sugar, and you get what happens is the NF-kappa B is hyper-oglycnaculated, it's over-modified. And what we expected to find the exact opposite. So we did an experiment where you did immunoprecipitation of NF-kappa B from brain extracts, and then you do Western blots to pick up the oglycnac-modified form and then the total form. And you just measure the ratio. So what we were surprisingly to find is that actually the NF-kappa B is key molecule for Alzheimer's, for inflammation. It's actually over-oglycnaculated. This could actually be pathogenic as opposed to protective. So my earlier theories about what was going on could be false. So again, this is a lesson to know that it's good to know what's actually going on in the human brain as opposed to what you might be able to reproduce in a culture dish. So in the last couple of minutes, I wanted to just sort of focus on a topic which is very dear to my heart, is basically, do we know what the microglia in the human brain are doing? The marker for it, widely used is either HLA-DR, the MHC you saw the picture earlier, or the IBA1, which basically stains all microglia. But when you try to subdivide them, it's really difficult to do with just these two markers. So we started to explore the idea of, is it possible that you have a subphenotype of different types of microglia? So we sort of used panels of antibody in different brain sections. It was interesting, CD14, this is known as the lipopolysaccharide receptor. There's been a couple of papers, but it's not widely used to look at human brain, and we think actually that might be a mistake. So CD14 probably is a classical M1, so cells that are overexpressing CD14 are probably more likely to be doing damage. And so in the normal brain, you can see it in the monocytes in the blood. But we also were able to see a lot of cells. These are double stained. For the purple is the CD14, and the brown is the IBA. I have to apologize. Normally it's traditional to show confocal pictures, but human brain is very difficult to work when you're looking at fluorescence because it autofluorescence strongly. So we've gone back to the old traditional methods. The other feature that we had was that the M2 marker, the classical marker, CD206, was not seen in, I've never seen it on a microglia, on cells like this. You see it strongly in macrophages and vessels. So this, again, highlights the need to be able to not just look at a tissue homogenate for Western blot, but actually to see the localization. We've done PCR or brain extracts, and you get a strong signal, but you don't see the protein because it's probably all coming from the blood. Now, I have spent a little bit of time trying to understand new markers here, because we really don't know what are the activated microglia, so the new markers. I'm just going to present two things. One is, can you define the microglia as being activated better? So by chance, we were looking with an antibody to endoglin. Now, endoglin is considered a vascular marker. And so you buy the antibody, you spend your $400, and you use it on the tissue sections. And what happens is that if you look at a brain section, you can see a lot of microglia, but you see one cell. This is an ND case that's got endoglin staining. This seems to be a subtype of this particular protein. This antibody doesn't react with the vascular cells. And then when you look at, in an AD case, where you expect to see more activated cells, you see more endoglin staining. Now, this study is still under investigation. I think what we were also seeing was if you actually do stain for endoglin and vessels, you might be staining for a subtype of possibly perivascular macrophages, ones that have come from the blood. But this is one way we're trying to see if we could sort of better identify activated microglia. And this also works in Parkinson's disease. You can see basically in areas where there's a lot of pathology, you get stronger endoglin staining. Again, this makes little sense biochemically. We're still under investigation, but it's kind of exciting to see when you do a tissue stain to see that what you're looking for, because the information you can get out of an HLA-DR stain, is sort of just unidimensional. Now, this is the final thing. This was just from last week, actually. As I mentioned earlier, one of the markers for looking at microglia in the brain, or defining microglia in the brain, is P2RY12. This has been defined in high-profile studies, both in human and in rodent. So you can see in both cases there's a huge difference of expression in the microglia. And we showed that, as I said earlier, in our studies. So when you stain the sections, you actually turn out, if you have the right antibody, it's a very interesting pattern of expression. These are sort of microglia that are very closely associated with the neuron. You can imagine they are the good microglia doing their job of looking after the neurons. And so some people have defined these as alternatively activated microglia. That's still a question to be answered. But what we were particularly interested in was that we believe that these are marker for unactivated microglia. And when the cells become activated, you lose the staining. This is an example. So you've got P2RY12 microglia, and you've got an HLA-DR. They really don't co-localize all that much. There's a little bit here and there, but you can see. So here you can actually define, what might be, in a better way, a more activated microglia from a normal microglia. I think the concept behind this is the idea that maybe all cells, all the microglia in an Alzheimer's brain are activated. They're not. And so here we have this picture where you've got the plaques and you've got the P2RY12 microglia, and they're not on the plaques. In fact, they're shying away from the plaques. This is a higher magnification again. You can see a faint remnant of a microglia stained, but most of them are away from the plaque. And this is in contrast to this one, where you've actually got the HLA-DR and you've got the plaque, the microglia on the plaque. Here, the P2RY12 are actually off the plaque. So these are sort of two different populations of microglia. So we're still kind of wondering whether, one has to assume these are non-activated microglia and they actually may be doing a good job. The literature is a little confusing about that, but it's sort of a new finding. And again, I think the idea of sort of having two populations of microglia in an Alzheimer's brain, being able to distinguish the non-activated from the activated is quite exciting, and it has potential for sort of, when you start to apply sort of more mechanistic approaches to these studies. I really sort of, this is kind of a bit arrogant, the summary, but it's basically 30 years of work. So, I mean, there have been many, many studies of neuroinflammation in Parkinson's and Alzheimer's for 30 years, but the hope of anti-inflammatory therapy right now has not been realized. We all have to do a lot more work to find new targets, because whether it's anti-inflammatory therapy or other therapies, most clinical trials are failing. And so, like with the researchers here, we're all looking for new anti-inflammatory targets. But I think this depends on having a fuller description of the inflammation associated with the brain disease. So that's been my little niche study. So although it's not favored amongst granting agencies, we're trying to sort of preach the idea that descriptive human brain pathology studies should still have a place in any scientific investigation of neuroinflammation. This was a picture of an Arizona sunset out of my office window before I moved to the office, but we have a group of highly qualified people and collaborators over the years who've contributed to the work, particularly my colleague Dr. Liu, who've shared a lab and done all our microglia together, and Dr. Tom Beach, who is the director of our brain bank, who is a fellow Canadian, and he actually hired me in 1998, which I'm very grateful for. So thank you very much.
Studies that show strength in the vicinity of the plug, still it's not clear whether it has protective or a beneficial or a toxic role. So my question is, has anybody studied P2Y or 1, 2, like what is the relationship with 1, 2 and also the plaque here?
No, I mean, I think we're going to have to do that. The literature on this is, there's been basically P2RY12, I publicated over it as an ATP and ADP receptor, purinergic receptor. And so the idea is that if you got release of ADP or ATP, whether it's from damaged cells or maybe normal function, it can activate the microglia or activate the receptor, and you get a calcium response. But whether that's pathogenic or not, the literature is a little discordant on that. Some people have said if you throw in like 200 micromolar ADP, you get increased TNF. But again, that doesn't, based on the observations I made, I don't think that makes sense. I mean, an earlier Nature study, which first sort of looked at this, said you take LPS and stick it into a rat brain or a mouse brain. You lose, again, the same thing is that the P2RY12 expression disappears. So it's like the conclusion is that you have a marker for non-activated microglia.
So you mentioned that GDP have we tried mapping out that gene to see if there's something clinically response in a mouse model or something?
Yeah, that was the earlier work. My competitor at Harvard, who got great resources, they developed a CD33 knockout mice. And their take on the thing is that if you've got the polymorphisms in the gene, it actually affects amyloid phagocytosis. And so they were sort of focusing on that type of study. And they show if you actually knock out the gene, you're going to affect how the microglia phagocytose the amyloid. They didn't actually look at, they were viewing it as a phagocytic receptor, which I think is wrong, because essentially CD33 binds to sialic acid modified proteins, it can activate an anti-inflammatory system that could basically maybe turn off a phagocytic receptor, but it also has other anti-inflammatory effects. Again, there's some interesting lessons from diabetes. So if you actually knock down CD33 or you reduce CD33 in diabetes, you make the inflammation worse. So again, as a therapeutic target for Alzheimer's disease, one has to be careful.
You said earlier in the presentation that Parkinson's disease has similar microglia activation, but Lewy bodies don't correlate with CD200. Do you think Parkinson's has its own
Yeah, I think, again, it comes down to the signaling molecules. Now, in vitro systems, many people, and we've done it as well, if you do an in vitro experiment or an animal experiment, you can show microglia can be activated by alpha-synuclein aggregated forms in a very similar manner to the amyloid experiments that people have been doing for years. I think that the concept is that the alpha-synuclein is driving the inflammation in Parkinson's disease has to be questioned because, as I said, I focus my hypothesis based on what I see in the brain and talking to pathologists. And my pathologist friend kind of agrees. You don't see this large inflammatory response to the alpha-synuclein in a human brain. Now, there's many caveats to that comment as a good scientist, because the alpha-synuclein could take different forms, which some of them we might not be seeing. So if you've got overproduction of like oligomeric alpha-synuclein, which might not be phosphorylated, being able to pick up phosphorylated forms of alpha-synuclein is a nice tool. And it's sort of, but it's sort of in terms of the progression from normal synuclein to this phosphorylated garbage dump, which is a Lewy body, you've got several intermediate steps. So we were just presenting the idea of, there seems to be a difference between this Lewy body inflammation and Alzheimer's pathology inflammation, but what it is, I don't know.
Regarding the naphthocytin of naphthocytin capsules, did you say there's a correlation, positive correlation between chemistry expression?
No, sorry. What we were actually using that slide was to show positive correlation between CD200 and synaptophysin. So what we actually, this kind of was just used to highlight the effect that the more CD200, the more neuroprotective it is doing, basically preserving the synapses. It's sort of, when you're mining sort of gene expression profiling data, you just see, you're seeing a correlation, a good strong correlation, but the cause and effect, again, has to be proven. But I think there's other data to support the idea from animal models that, if you lose the CD200, you're going to get enhanced neuronal cell deaths for some reason. So again, we were just sort of mining human data to support our hypothesis.
Let's start the discussion. We'll be available in a few minutes if you want to talk more. Also, the classroom. The class is going to start soon. So then thank Dr. Walker again for the talk.
Thank you.
Thank you very much.
Okay.
Fantastic.
Okay, good. Could I be heard?
Oh, it was good. It was good. Okay.